I see Heather.
Yeah, hi
Heather.
there. Sorry.
Yeah.
It's who is recording? Is it a participant or the host?
I still have some people in the office. Sorry, I'm just coming out of a meeting at work. So my name is Heather. I'm a science teacher at UNIS, the United Nations International School in Manhattan. And I didn't get the first part of the question because I was just coming in, but I think it was who we are, where we work, and then
Yeah, what maybe what
here.
grade level and subjects you teach,
Yeah.
and then why you signed up for this workshop.
Yeah. So right now it's general science, and I was teaching IB bio before. And then what brought me here is just like I want to be able to try to get my kids out more to do some ecology work. So I wanted to be able to learn just like the skill set and sampling, looking at quality water. And at my old school, we used to actually work with the Department of Education to the Day in the Hudson, where we would sample water. So just kind of upping my skills again and refreshing my memory on how to do it. And a long time ago, I can't remember which summer, but I came out one summer to help collect, just do some work with some oysters, just like in measurements, counting the richness of them. So that was really fun. So I would just love to kind of continue learning what BOP is doing and maybe looking at even in the future, trying to bring it into the classroom. Again, my old school has a tank right now with oysters in them because we used to work with, we were working with BOP, but I'm at a new school now. So I'm trying to maybe implement into this new school as well.
Great, thank you, Heather.
Thanks.
And then Salima, could you please just introduce yourself, school, grade, subjects, and then maybe just why you signed up for this. Okay, maybe there was a bad connection there, but okay, so we lost Salima. Okay, so we'll just we'll hold on. We have another person joining, Amy. Hi, Amy. We're happy to have you join us. I'm Brett Franco from Brooklyn College, and we were just doing a quick round of introductions. So we'd love to hear where you teach, what subjects, grade level, and then why you signed up for this workshop.
I teach at New Explorations into Science, Technology, and Math, or NEST+M. I teach science to 3rd and 4th grade and technology to 5th grade. And I signed up, well, for a number of reasons, including, it ties in directly with both science and technology. And especially the technology component of my classes is very much an evolving thing, because the students have had experience, typing more than enough to last them for a little while at least. So my goal is to make sure that the curriculum is dynamic and responds to the changing world, as well as introduces new things. So learning about the science technology integration that is part of, testing water and responding to what those results are is directly where what I want to teach into. It makes them both responsible citizens and gives them the content area that I'm responsible for, to my mind.
Great, thanks, Amy. And have you done water quality testing before of any sort?
Do I need to say yes? I haven't.
No, you don't. You absolutely don't. I'm just asking to just so I can understand sort of like where everyone is coming into this discussion.
I generally like to know what other people have said first, and since I got home late, I don't get to cheat.
Yeah.
And I don't have any previous experience, but I'm always willing to play along, because I also always am interested in my own edification if it's not something I can apply.
Right. That's a perfectly fine answer, and so I'll just jump right into it. So what I wanted, it's a little frustrating that we have to do this via Zoom, right? Because I think the original vision for this was that it was going to be an in-person training alongside the water, but so be it. So we'll I'm going to do my best to at least make it dynamic. I won't show you very many slides with words on them. And I will try very, very hard not to talk too much. But what I would like to do today is introduce the idea of nutrients as a water quality issue. So we'll talk a little bit about nitrogen and phosphorus. We will talk a little bit about. I'll introduce you to some of the methods that are used to measure nutrients in surface waters. I kind of went back and forth about how much I wanted to like demonstrate that, because I personally feel there's very limited use in showing someone on Zoom how to do something if they can't be holding everything in their hands. But I did bring some, some actual water quality testing kits with me, so I might demonstrate one briefly, and then I really want to talk a little bit about or not talk, go through an exercise so that we can talk about the context. When you get a water quality measurement, what does it mean? And how do you interpret it, especially given the limitations of what we typically do in a classroom setting where we're limited by time and money and available technology. So I'll show you a useful tool online, and then we can we'll conclude with you just telling me what you found useful in this hour and a half, and what you might be able to bring into your classroom, and then that might prompt me to follow up with some other resources for you later. So that's kind of the game plan. It's a small group, so please jump in at any time. This isn't meant to be a one-way communication. Okay, I'd love to engage the three of you in discussion and answer questions and see where your interests take you, because it's always interesting to see where people want to go when you're presenting something like this. So without further ado, I do have the obligatory PowerPoint to start anyways. So first, a very quick about my background. So I'm a marine scientist by training. I went to the University of Connecticut as both an undergrad. Actually, as an undergraduate, I spent a lot of time in the summer working for a professor. We would sample the water quality at all these different little bays and inlets around southeastern Connecticut and Rhode Island as part of a project to understand the ecology of these systems. So I spent a lot of time on the water doing these water quality measurements. I stayed at UConn to work on my PhD and became interested in the water quality problems of shallow lakes. This is actually a small shallow lake on the main campus of the University of Connecticut, and we developed all kinds of new tools and equipment for sampling waters that were only on the order of 6 feet deep, and trying to get the fine scale measurements of how things like dissolved oxygen and nutrients change as you move from the surface down to the bottom in only 6 feet of water. So we did a lot of technology development there. And then I went and did a postdoc at the University of Western Australia, where we're looking at the movement of water and the dissolved constituents of water, like nutrients and things like that, through underwater canopies, meaning like seagrass forests and things like that. And then I came to Brooklyn College and set up some water quality testing programs in Prospect Park Lake. Prospect Park Lake has some severe water quality issues. They get intense algae blooms that are triggered by the input of phosphorus. So we spent a lot of time working and helping to define that issue better for the Prospect Park Alliance. And I've done a lot of water quality work in the waters around New York City. This is actually a photo from off of Soundview in the Bronx, where there's actually a very active oyster restoration project going on now. And so, I've had a lot of experience working in freshwater, saltwater, estuaries, et cetera, on water quality issues, and using a lot of different techniques and trying to develop lots of new sort of like methods for measuring that. So that's just a little bit about my background. So what we're going to do today, our objectives for today, we're going to discuss very briefly. I'm going to go through some of this background stuff very briefly. The importance and impact of nitrogen, and specifically we'll zero in on nitrogen, because that's the most important nutrient for for the waters around New York City. So we'll talk about the importance and impact of nitrogen. And I want to discuss a little bit about New York State standards, because usually when we measure water quality, we're doing that in reference to some sort of standard that's mandated by the Clean Water Act. It's how we determine whether the water quality in a water body is good or bad by comparing it to some sort of standard. So we'll talk very briefly about what it is for nitrogen. I'll describe some basic methods for measuring nitrogen, particularly ammonia and nitrate, and the strengths and weaknesses of each. Oh, geez, this says dissolved oxygen. It should say nitrogen. We recycled the presentation from the dissolved oxygen session, but we'll talk about the factors that control nitrogen concentrations. And then we'll, like I said, I have an exercise where we can start to think about interpreting spatial and temporal patterns. How does nitrogen concentration change with location and with time in the waters around New York City? So that's sort of the game plan. So first, has anyone ever seen a figure like this before? This is a pretty standard sort of like textbook diagram of the nitrogen cycle in natural waters. I see Heather shaking her head yes. Amy, have you ever? Okay, Amy's not seen this before. And I'm sorry, it was Catherine, right? Please say I got that name right. It's Catherine Sweat, right? Okay. So anyway, so the nitrogen cycle, like I said, this is a pretty standard one, and it's the thing I want you to get out of this brief discussion is that there's different forms of nitrogen, right, that are important in waters, and there's an exchange of nitrogen between the water and the atmosphere. So let's start right here with nitrogen gas, right? The atmosphere has a lot of nitrogen gas, and this can enter the water through the activity of bacteria, where it's converted from nitrogen gas into ammonia. And ammonia is a nutrient, right, that can be used for growth, right? So it's needed to build organic matter, and it starts with plants and other types of photosynthetic organisms. So we can have this transfer of nitrogen from the atmosphere into ammonia, and then through various different types of reactions, most of them facilitated by bacteria, that nitrogen can keep changing forms. It can be turned from ammonia into nitrite, which is this NO2. It can be turned from nitrite into nitrate, which is NO3. And then there's other types of bacteria that can take nitrate and turn it right back into nitrogen gas and send it back to the atmosphere. So that's sort of like like I said, sort of like a very simplified version of what a nitrogen cycle might look like in the world. And a couple of things in this diagram are missing. And the big one is organic nitrogen, right? Because in this view, right, we see ammonia, this NH3, we see ammonium, the NH4 plus, we see nitrite, NO2 minus, and nitrate, NO3 minus. We see those as just sort of maybe molecules that are floating around in the water, like dissolved forms of nitrogen. But remember that there's biology happening, and organisms are taking up these nutrient forms of nitrogen and converting it into organic nitrogen, right? Incorporating it into the biomass of the living earth. And so there's a big pool of organic nitrogen too. And that's important to remember, because typically if you go in and you measure nitrogen, you're typically measuring ammonia, nitrite, and nitrate, and it's only a partial picture of all the nitrogen that's in the environment. You're not typically getting the organic component of the nitrogen. Okay. Now, this again, I said is a very simplistic view of nitrogen. A more realistic view of nitrogen for places like New York City might be more like this. And in this picture, what I want to convey is that rather than the major input of nitrogen into our waters being from this transfer from the atmosphere to the oceans, most of the nitrogen that enters the waters around places like New York City are coming through wastewater, wastewater, right? The stuff that's going down our toilets and sinks, et cetera. And that usually flows through our sewer systems. It may or may not get treated, and then it's typically discharged into the waters around New York City in various forms. It can be organic nitrogen. There might be lots of ammonia. There might be some nitrate, but there's an awful lot of organic nitrogen being discharged. And that organic nitrogen then enters the nitrogen cycle. It can be taken up, incorporated into biomass. It can be broken down, turned into ammonia, nitrate, nitrite, et cetera, right? And so, this is, the most significant input of nitrogen into the waters around a place like New York City. Generally speaking, where does nitrogen come from? What are other sources of nitrogen into water bodies, right? And we usually classify sources of nitrogen as either point or non-point. And point sources are those sources where you can literally go and find like the end of the pipe from which the nitrogen is coming, or a very specific discharge point where it's easy to locate it and measure it, for example. And examples are like sewage treatment plants, the discharge from sewage treatment plants, which is a big one for New York City. It can come from agriculture, animal feedlots. There's these massive, for example, pig farms down in around the Maryland area that discharge large amounts of nitrogen waste directly into the water. It can be industrial waste. And then you have these non-point sources, which are a little more diffuse. The nitrogen's kind of coming from everywhere. And that can include from mining operations. It could be fertilizer running off farmland. It can be coming from construction sites, and it can even come from smokestacks. There's a lot of nitrogen in gaseous forms that's discharged through combustion, and then it kind of rains down onto the water bodies as wet or dry deposition. So those are sort of examples of non-point sources. And what are the consequences? So again, I'm kind of going through this in a very simplistic way, but the interesting thing about nitrogen is that at its core, nitrate, ammonia, those forms of nitrogen are critical for life, right? Without nitrogen, without phosphorus, without sulfur, without these basic building blocks of organic matter, there would be no biosphere. So on the one hand, nitrogen is a good thing, right? Because all life depends on having a supply of nitrogen to create biomass. I mean, a lot of our body is made out of nitrogen. It's the fourth most abundant element in life, behind carbon, hydrogen, and oxygen. So why can it be considered a bad thing? Well, because if it's in excess, if there's too much nitrogen, you're basically over fertilizing the environment, right? Just like I live out on Long Island where people are just like crazy about their lawns, and the way that you keep a lawn thick and lush and green is you constantly apply fertilizer to it, and that's giving the grass basically lots of nitrogen, phosphorus, and potassium to grow. And a similar thing happens when you put large amounts of nitrogen into the waters around New York City. It stimulates growth, primary producers. And they might not always be the primary producers that we would consider to be preferable. So in this diagram, I'm showing what a sort of classic picture of what a healthy bay looks like, right? Which is water that's fairly clear. The sunlight can penetrate down to the bottom. There might be seagrass meadows down there, which can receive that light. There's some nitrogen around so that they can grow. And that seagrass communities can support a rich biodiversity of other marine organisms, including fish. But what happens when you start to increase the amount of nitrogen flowing into a bay is that you stimulate the photosynthesis of other types of primary producers. for example, seaweeds, right? Seaweeds, and I'll show you an example of that in a second, or single-celled organisms called algae, or actually even some, there are some single-celled photosynthetic bacteria as well, or maybe even free-floating plants like duckweed in Prospect Park, for example. And what happens is when you stimulate the overproduction of these other types of primary producers, they start to change the ecosystem quite a bit. They can start to shade the water. They reduce the penetration of light because they're intercepting that sunlight, and so you can no longer support seagrass, which is considered to be a very preferable type of habitat. You can also, a lot of those single-celled organisms that can grow in great abundance when you have too much nitrogen can be toxic. So you have to worry about stimulating what's called harmful algal blooms, right? Blooms of toxic single-celled organisms that are so thick that they color the water. It might look pea soup green, it might look red, it might look brown, it might look mahogany. And people refer to these as green tides, red tides, brown tides, mahogany tides. The colors are coming from the pigments contained within the algae. And some of those tides, the red tides, for example, again, can be toxic. So that means that there's a human health problem. Those toxins can be taken up by things like shellfish. And then if you consume those shellfish, you can be exposed to the toxins. And it might be harmful to some marine life. And then the third biggest problem that happens when you stimulate this overproduction, this fertilization, is that eventually all that stuff that grows and blooms will die, right? Just like the trees lose their leaves in the fall and the grass turns brown. A lot of the primary producers in the marine ecosystem will start to die off in the late summer and early fall, and all that dead stuff falls to the bottom of the water body, and it decays. And when it decays, it sucks all the oxygen out of the water. It creates these low oxygen environments, which can then cause fish kills and things like that, or dead zones at the bottom of these water bodies. So, so these are some of the consequences, right, of too much nitrogen in the system. And that process of putting too much nitrogen into the system, creating an overabundance of algae or microalgae, it's called eutrophication. And I realize now I didn't put that word anywhere, so I'm just going to type it into the chat. It's an important word in my business. Eutrophication, right, is basically an excess of nutrients stimulating an overabundance of primary production. And then the consequences are the ones that I mentioned: harmful algal blooms, low oxygen conditions, fundamental change of habitat, loss of biodiversity, et cetera. So, so this is why we're concerned about how much nitrogen is in the water, right? We want some, but not too much. And so we routinely measure it so that we can track the amount and decide if we need to do something to intervene and reduce the amount of nitrogen that's in the water. And just to drive the point home, this is just a diagram or a graph showing what some of the leading causes of impaired surface waters in the United States are. And when I say the word impaired, what I mean is, the Clean Water Act. The Clean Water Act requires that the surface waters of the United States be fishable and swimmable. That's sort of like the standard, right? Fishable, swimmable waters, and things that and to be swimmable, right? It has to not be harmful to human health. So so impairments are things that that reduce the or prevent a water body from being fishable and or swimmable. And so some of the leading causes of impairments are the presence of pathogens, typically from raw sewage, the presence of heavy metals, typically from some sort of industrial process like mercury and lead, and nutrients are number 3. So again, it's an interesting paradox, right? Nutrients are necessary to support life, and yet in excess, they can prevent us from reaching our goal of fishable, swimmable waters. Now, this is a photo from Jamaica Bay at low tide, and I include this in here just to show you an example. So Jamaica Bay has an overabundance of nitrogen, and it helps to promote the growth of lots of seaweed in the summertime. You get these vast beds of seaweed, in this case, different types of ulva species. So at low tide, you can, the entire mudflats will be coated with these layers of seaweed. And when they decay, again, they can consume a lot of oxygen, and they can promote, they produce a lot of hydrogen sulfide gas, so it gets really, really smelly out there when this stuff starts to decay. And just here's another photo from one of my former graduate students showing what some of this algae looks like that grows in Jamaica Bay. So again, Jamaica Bay is a system that we could say is eutrophic. It has an overabundance of nitrogen, and a consequence of that is that we get lots of growth of things like seaweeds and single-celled organisms that can make the water look green or brown. And then I'm just going to conclude this sort of section by just talking about what the state standard is for nutrients in water bodies. So one of the challenges about trying to regulate nitrogen is that it's really hard to pinpoint a specific concentration of nitrogen in the waters that will create the sorts of problems that we try to avoid. And therefore, New York State does not have a specific standard, a specific measurement of nitrogen that we need to stay below, right? Rather, they have what is called a narrative water quality standard rather than a numeric standard. And narrative standards just describe the conditions that we are trying to achieve, right? And so, so the narrative standard for nitrogen is that it will be low enough that we don't promote the growth of algae, weeds, and slimes that will impair the waters for their best usages. Nice and vague, right? And so really what we're trying to do is we're trying to reduce nitrogen to the point where we'll no longer have these massive blooms of macroalgae, for example. Exactly how much that is, that's an active area of research. It's why we have to constantly monitor the amount of nitrogen in the water, and also other parts of the ecosystem, so that we can see how it responds to different levels of nitrogen, and that helps us determine whether or not we're meeting the standard or not. Okay, so I'm going to pause there. I'm going to stop sharing for a moment to see if anyone has any initial questions. Was that too fast? Was that too much info? Is there anything that you'd like to talk about a little bit more? So any questions or comments or anything? Yes, please, Heather.
I'm interested in like the very vague standard they have for that narrative. It's like best usage. So does it mean that throughout New York State, there's different levels that you could be with because the usage of the water is different?
Yeah. The way that they, it's really a complicated problem, right? But so first of all of New York State waters have a very specific classification given to them by the state, which determines what the state considers their best use. So some waters, there's like class SA, class SB, class SC. There's all these different classes. Some of them are about what they call primary contact recreation. Some of them are about healthy fish habitats, et cetera. So that's one problem, right? Is that different waters have different standards. But because these water bodies are all different, some are big, some are small, some flush very quickly, some the water stays in there forever. They respond differently to different amounts of nitrogen. And one of the ways that we try to regulate nitrogen is, rather than regulate how much is in the water, oftentimes we try to regulate how much we're putting into the water. we try to regulate the amount of nitrogen that can be discharged into the water rather than regulate the concentrations themselves. And that those standards are typically based on what the scientists think the water body can handle, right? Based on like research and monitoring over long periods of time and trying to understand how the system reacts. So every water body could end up having a different standard for how much nitrogen you can put into it based on its characteristics, its classification, and how we think it works. So it's quite complicated. Other questions? Does that answer your question, Heather?
Yeah, I was gonna say, hence why they keep it so vague. Sorry.
Yeah, that is why, you know, because they don't want to put impossible restrictions on this for people, right? Unachievable restrictions. Other questions? That's a good question. Any others?
I just wanted to share. Hi, it's Cynthia. I just wanted to share that I used to work with the wastewater treatment plant that was discharging into some marshlands in the Bay Park area in the South Shore of Long Island. And at that location, because of the really sensitive marshland, it was decided that at that particular area, the marshlands couldn't take any nitrogen inputs. So there's also situations like that, depending on, you know, the ecosystem that places are discharging from. Yeah.
Yeah, so you totally understand the difficulties in trying to figure out, you know, how much nitrogen is too much, right?
Yes, there's a lot of back and forth and just many reports with the state agencies. Yes.
Yeah, absolutely. Okay, well, so let's transition now to so this is why nitrogen is important, and so let's think about now, how do we measure it? You know, keeping in mind that I just, you know, when we started with the nitrogen cycle, there's many different forms of nitrogen, and so our approach to measuring it is different depending on whether it's nitrate, or whether it's ammonia, or whether it's organic nitrogen. And I'll try to briefly go over it. I'm not gonna go in depth into the principles of operation of all these different methods, but I'm gonna give you a sense of what kinds of options are out there, how expensive they are, and what some of the pros and cons are. And we'll end with some things that you're most likely to be able to access in a classroom setting. Okay. So back to the PowerPoint. So we have to measure nitrogen in our water bodies, and just like in any field of research, the more information we can get, the better. And so I'm starting by showing you two ways that we can measure nitrate and ammonia in water bodies, and in ways that allow us to collect you know, lots of information over long periods of time. So these are sort of like automated methods. So the first is these ion selective electrodes. These are basically what we could call electrochemical methods, right? The basic idea is that both ammonium ions and nitrate, you know, they're both ions, and so that they can interact with metals, and that interaction oftentimes can produce a current. And we can typically measure that with some type of current meter. So, so there are ion selective electrodes available for both nitrate and ammonium. But the problem with that is that I've actually used them before, and I found them to be very unreliable in marine systems. They foul very easily. They're very difficult to calibrate against known concentrations. There's lots of things that can interfere with them. So if there's other ions in the water, it might interfere with the operation of the electrodes. There's just a lot of problems associated with it, but they actually work very well in industrial applications where conditions are a lot more controlled. So they're available, they're out there. I don't use them, and I don't know very many people that do. A state-of-the-art way to measure nitrate in particular is this instrument right here. It's manufactured by a company called Seabird, and it measures only nitrate, right? Only that one specific form of nitrogen, just nitrate, and it measures it by passing an ultraviolet beam through the water and measuring how much of that ultraviolet radiation is absorbed, because nitrate is a very good absorber of ultraviolet radiation. So you can put one of these instruments out. They're battery operated. You can put it out in the open ocean for months at a time and take measurements every 15 minutes. But some of the issues associated with this is, number one, it can only measure nitrate. It can't measure ammonia or organic forms. It's a very expensive instrument. Instruments like this can cost $30,000 or so, and that's just for the instrument. And then it requires frequent maintenance and cleaning and things like that. And it works very well out in the open water, out in the open ocean, where the water quality is generally better. In environments like around New York City, where there's a lot of where the water quality is a lot more poor, the water's murky, etc. It doesn't work as well. But this is sort of like the state of the art for measuring nitrate in open waters. There's a lot of these out in the open ocean. But the tried and true methods for measuring nitrogen, I always like to think of these as like the old-fashioned way, because some of these methods have been around, since the early 20th century, is through a process called colorimetrics. And the way colorimetrics work is you take a water sample and you add some reagents to it, reagents, which is just other chemicals, which will react with the nutrient that you're interested in. So, for example, if you're interested in ammonia, you can add reagents, chemicals that will specifically bind with the ammonia and produce a dye. In this case, for ammonia, it's called an indophenol dye, and it's blue. And the more ammonia that's in the water, the more blue the dye. You'll create more dye, and the more blue the sample will get. And like I said, it's a very tried and true method. And I'll explain in a second that there's lots of variations on this. There's also a colorimetric method for nitrate and nitrite. You can add chemicals to a sample that contains nitrite, and it'll create what's called an azo dye, which turns pink. And again, the more nitrite that's in the water, the pinker the sample will get. And based on those two very basic, chemical methods, right? The one for ammonia and the one for nitrite, we can think of ways to measure all the other types of nitrogen that we're interested in. I see Salima said that she's that they're familiar with these colorimetric methods. Is that right? Is that what you were saying? You've used kits like this before, right? And so we can build on these. So, for example, remember I said something earlier about organic nitrogen and how the water can have a lot of organic nitrogen in it. And if we want to measure how much organic nitrogen is in there, the way we do that is we first process a sample. We do something called a digestion. We'll add, some really strong acids and some strong oxidizing agents, and we'll try to break down all the organic matter and basically turn it into ammonia, and then just measure how much ammonia is there. And so there's a very specific method for that called total. The measurements named after the person who invented it, some scientist with the last name of Kjeldahl, So there's this method you might see called total Kildall nitrogen, which is basically taking a sample, adding these strong acids and oxidizers, trying to turn all the organic nitrogen into ammonia, and then using this very simple colorimetric method for measuring how much ammonia is now in the sample. Similarly, if we want to measure nitrate, we can take this first step of turning all the nitrate into nitrite, and then we apply that very simple colorimetric method to create the azo dye, and then we can measure how much nitrate's in the water, right? So these chemical methods are the tried and true, and we have a lot of variations that we can build on top of these very basic measurements of ammonia and nitrite. Okay. Now, once you have the dye, and again, the color, how deep the color is, how intense the color is going to depend on how much of the nutrient, how much of the ammonia, or how much of the nitrite is in the water. And there's in that color, the amount of dye produced is basically proportional to the amount of nutrient that's there. So how do you measure it? Well, the most basic way is to compare the color that you get in your sample with some color standards. And this is the cheapest and simplest way to do these colorimetric methods. And so literally you can buy a kit for like $100 from this company called Lamont, and they'll give you the reagents to create, that you can add to your samples. And then they give you like a little comparator. It looks very much like it does in this slide, right? It's just a series of another series of tubes, and what you do is you hold your sample up against that. You try to compare your sample to one of these sort of standards, and then simply read off the concentration. So it's a very simple way to do this. The only problem with it is that it can be it's not very precise, right? The steps between the concentrations can be on the order of 1 milligram per liter or 0.5 milligram per liter. So your resolution isn't very good. And also, the color can be very difficult to match up, and it can vary from person to person. Every time I've ever tried to use one of these, you got like 5 people saying, I don't know, what color do you think it is? Or I think it's this one, and someone will be like, no, I think it's this one. So it's very open to to interpretation. So it's not very accurate, it's not very precise, but it can give you a general idea, right? It can give you a general idea of whether your nutrient levels are high or low. They're actually very useful if you have an aquarium in your classroom, right? And you're trying to maintain, let's say you're trying to maintain your ammonium, your tank kind of low. Kits like this can be very useful just to know if you're, if you're in excess or not. Right, so here's just some of the, this is just summarizing what I just said, right? They're very easy and quick. However, they're very subjective, and the colors can look different depending on the lighting, depending on if there was if the water was murky to begin with. So they do have some limited value from a scientific standpoint. Now, the other way that you can measure how much dye you created in this reaction is through a process called filter photometry. And I'm going to take a minute to explain this because this is really the way that most people, when they do research or are measuring nutrient levels in a water body, ultimately determine the concentration of the nitrogen. So the basic way that a filter photometer works is you start with a light source, just some kind of bulb, and you pass that through some apparatus, which in some cases can filter the light down to a very specific wavelength. And I'm going to show you a different picture in a second to sort of explain that, right? But we take essentially white light, like every light bulb puts out essentially white light, and we try to filter that down to a very specific wavelength of red light or green light or blue light or something like that. Because each one of these dyes that's produced in these reactions absorbs light in a very specific wavelength. So for example, I said that nitrate produces something called azo dye, which looks pink to your eye. And it absorbs light very well at 520 nanometers. That's the wavelength of light. The 520 nanometers, which is getting down towards the blue end of the light spectrum. So it looks pink to your eye because it doesn't absorb red lights very well. It reflects them back to your eye, but it absorbs blue light very well. So when we want to measure nitrate in the azo dye, we try to filter down to 520 nanometers. And we take that filtered light and we pass it through the sample and see how much gets to the other side, right? And so the more dye that's in the water, the less of that very specific wavelength of light will pass through to the other side. And we can measure, right? We can measure how much light we sent in, and we can measure how much light comes out. And from that, we can get a signal. We can get an electrical signal, and we can calibrate that to a concentration of the nutrient in the water. So it's a very, it's basically replicating what you're trying to do when you use the visual comparators. You're trying to judge how much color there is in the water, but when you use a filter photometer, it can do that much more precisely because it's literally measuring how much light can pass through a sample, and the amount of light that can pass through the sample is proportional to, or inversely proportional to, the amount of dye in the water. And here's just another picture kind of showing the same kind of setup, right? Light source, a way to split that white light into the different wavelengths, filtering it down to a very specific one, passing it through the sample, and then measuring how much light comes through the other side. And I just included this little electromagnetic spectrum here so you can get a sense for, right? For nitrate, we typically pass a 520 nanometer light, right, which is actually in the green part of the spectrum. So the dye looks more pinkish, reddish to your eye, but absorbs very strongly in the green part of the visible light spectrum. Similarly, the ammonia looks very blue to the naked eye. It can look very blue, but we actually pass a 660 nanometer wavelength through that sample, which is getting up towards the red end of the spectrum, right? So we pass a red light through that blue dye and see how much makes it through to the other side. So that's the basics, right? The basics is we add some reagents to a sample. It creates a dye of varying intensities, and then we measure how much light we can pass through it. Now, the way we can do that, the instruments available for measuring how much light can pass through a sample vary widely, and the price point on these instruments varies very widely. I actually have one of these in my lab. It's actually broken right now, but when it's not broken, it works very well. But this instrument is awesome because you can mix up reagents beforehand, and it's got a robotic arm that will mix all the chemicals up with your sample automatically, so you don't have to do that manually. And then it will suck those samples up one by one into a cuvette, right, which is just a receptacle to hold the sample that we're going to pass the light through, and then it shines the light through, and then it tells you what the concentration is. In this machine, I've really, so a machine like this might cost say like somewhere between $50,000 and $100,000, depending on how many samples it's sized for, et cetera. But I used to, like I said, I've used one of these in my lab, which was really useful because I could train an undergraduate to use this in about a week, and then they could operate this on their own, which was fabulous. They would have to mix up the reagents, right, to do the colorimetric part of it. They would have to learn how to program the instrument. They would have to understand, just some very basic principles, but then they could do this on their own. So this is one example of a machine that can do that colorimetrics. Here's another example. This one's actually really even a step up from that. This is called a segmented flow machine. It basically does the same thing. It mixes up all the reagents with your sample automatically, and basically just automates the whole process for you. And these machines can process large numbers of samples, so it has a high throughput. So most water chemistry, most professional water chemistry labs, where you're analyzing samples as part of, like, say, a monitoring program, will probably have a machine like this to run your samples. So you go out and you collect the sample and you ship them to a lab, and they run it for you. So these machines can be very expensive, over 100 grand probably, depending on what you get. And it requires a lot more skill and maintenance to operate. So so again, you'll find these in professional labs where you might have a support staff to help operate them, but not something you're gonna buy for your classroom. Now I'm gonna stop sharing for a second because there are much cheaper and simpler options, right? So so I have this little instrument right here, which is made by the Lamont company. This is actually a filter photometer. It comes with this little kit. So these are the reagents that you would need to add to a water sample to measure the amount of ammonia in there. So if I took a water sample, and if we have time, I might demonstrate this really quick, but if you add these reagents in the right order to a water sample and there's ammonia in there, it will turn it blue. And then you can take that sample. You would have it like in a little container like this. So this is the cuvette, and then you could plop it into this photometer, which has all the it has a filter to get the right wavelength of light, right? So your little thing can pop right in here if I had the right setting on it. You could pop it in there anyway. So this goes in this instrument, and you can measure directly, right? So there's a little handheld version of those much more complicated and expensive instruments that I just showed you. Now, how much is this? So this particular instrument, which would come with the whole nitrogen kit, you can get for $1,229. So still expensive, right? Still expensive, but starting to get a little more in the in the right range. Now, the nice thing about sort of like a pocket filter photometer like this is that they're very simple to use. The only thing that you really have to worry about is that some of these chemicals that you need to do the colorimetrics are hazardous. So you would need to really do this in a very controlled setting if you're doing with students. Well, if you're doing it anywhere, anyone needs to be careful, right? But you need the proper safety equipment. You would want to do it in a place, really that's controlled. But it's doable. Now there's other versions, right? There's other versions of this. There's another system that you could buy that could do. Well, let me back up for a second. The nice thing about this unit is that you can program it to do lots of different types of nutrients. So in other words, it can you can access lots of different wavelengths of light. So it means you can do nitrate, and you can do ammonia, and you can do phosphorus, et cetera, et cetera. You can buy simpler units that that only are specific to a certain type of test. So in other words, they can only do really one wavelength of light. And units like that are down. Now you're getting down into like the $600 range. So you could buy a unit that would be like sort of similar to this, but it could do, say, only nitrate or only ammonia. And then the other part, is that sometimes the kits, you have to sort of, mix up the chemicals, add the chemicals one by one yourself. There's other types of units where everything is already sort of like preloaded into a glass into a glass tube that's sealed, and you can just stick it down into water sample and snap off the end, and the water sample gets sucked up into the tube and automatically mixes with the reagents, and all the magic, all the chemistry happens right inside there. And then those tubes can get stuck directly into a filter photometer, so you can read how much light passes through, right? And so those units, again, you're looking at a few $100s to be able to purchase something like that. But once you have it, and every once in a while you would have to replenish the reagents, right? So there's a good question in the chat. Are you aware of any schools, colleges, labs that has a lending equipment program where classrooms can borrow for a few weeks, then return? I'll tell you right now that that I would be more than happy to lend this unit to a classroom for such a purpose. But I don't know of any sort of like general program like that. So that's a good thing to maybe follow up with BOP about. So Cynthia, I might follow up with you about that, because that's a great idea. It'd be nice to have these that you could lend out, right, for a couple of weeks that a class could use, and then return. I think I'm going to pause there and see if anyone has any questions or reactions to that, to anything that I've just presented. So again, just to recap, right? Colorimetrics are the gold standard for these measurements. You know, you can measure concentration just with your eye to see how intense the color gets, and kits for that can cost as little as $100. But really, to get precise about it and accurate about it, you really need to use filter photometry. A unit like this is more affordable, but again, it's not very, it's more accurate than using your eyeballs, but it's not as accurate and precise as the $50,000 and $100,000 instruments that I also just sort of showed you that you would get in a professional lab. So, okay, anyways, so anyone have any questions about any of that? Yes, please, Heather.
The colorimetric, looking at the color wheel, like if I was working with this with my students, and I know it's going to depend on the ecosystem you are in, the amount of water, body of water you're looking at, but what shade should I, like, should that be the conversation, like with the kids of like, that this shade, this would be at risk of like having too much of the nutrients in that body of water, that like it could then lead to an algae bloom, and then, you know, ultimately a eutrophication you know, of happening if that's in the water system. Just like looking at that color wheel, I'm not really sure like what shade I would talk to the kids about, because the kids will ask like, when is this dangerous? Like, what am I looking at? This is really light, this is really dark.
Yeah. Can you hold that thought? Because I think what I'm going to do next, which is, you know, doing a little bit of data exploration, might help to answer that question. You know, and I almost said, you know, it's complicated, but I don't want to make it complicated. It can actually be quite simple, right? But just to preview what I'm going to say, part of it is depending on you having a little bit of understanding of what kinds of concentrations are out there in the water body, and what kind of state the water body is in. Or if you're talking about an aquarium, I think there's more specific guidelines on the concentrations that are considered safe, right? Because too much ammonia in an aquarium can be toxic to the organisms in there, right? And that's a little more well defined than the concentrations out in a place like New York Harbor or Jamaica Bay that would be considered good or bad, right? But I'm going to show you some information that might help guide a discussion like that. Other questions? Originally, I had intended to take a break, but I'm realizing now that we're an hour in, and if we take a break, we won't have much time at all to sort of conclude. So, but still, at the same time, if people need 5 minutes to just kind of like recharge their brain, I know 50 minutes is typically like the time that people can maintain concentration, but Do people want to take 5 minutes, or do we want to sort of keep going for another 20 minutes?
I'm good if everyone else is.
Yeah.
Okay,
I'm good as well.
thank you. Thanks, Amy. Okay, so for this next part, I'm gonna drop a web link in the chat, and I would welcome you to feel free if you're if you're on a machine, if you're on a laptop or desktop, and you can sort of like see the Zoom and check out this link at the same time, feel free to do that. Or you can just follow along with me. You can just follow along with me on the Zoom. I'm just gonna walk through a little bit of exploration of some actual nutrient data from Jamaica Bay, New York. Okay, so I just dropped the link down in the chat. And I'm gonna just recommend that you don't get ahead of me in clicking around on this tool once it opens up, because we don't have a lot of time for me to like walk you through the whole tool. So I'm just gonna do some very specific things. Okay, so I think a really important thing, let me just give a little bit of intro to this. One of the things that I find, just in my teaching, and also in talking and working with high school teachers and middle school teachers in the past, is that oftentimes we spend so much time just going through the mechanics of doing the measurements and collecting the water quality data, entering it into spreadsheets, struggling to graph it in Excel or on Google Sheets or something like that. That we spend very little precious time in discussing what the data mean, and that's really the most important part of water quality measurements, right? Is interpreting what you're seeing. It's the only reason we make the measurements in the first place. So so I try to find simple ways to sort of like fast forward from getting a data point to jumping right into some kind of discussion about what it might mean. And in order to understand what it might mean, we have to have some sort of sense of what normal typical values are. So one of the problems with any water quality measurement is that you can get a different answer if you measure it at 6 o'clock in the morning versus 6 o'clock at night. You can get a different answer if you measure it in January versus June, and you can get a very different answer if you measure it from Canarsie Pier in Jamaica Bay versus off of Coney Island Beach. So you'll get a different answer depending on the time, whether it be time of day, time of season, or even one decade to the next. And you'll get a different answer depending on where you measure it. So one of the and a reason for that, right? And this is I think the part of the really understanding water quality. So for something like nitrogen, one of the reasons it might change depending on where you are is because you might be nearer or further from the discharge of a wastewater treatment plant.
Yeah.
Right? And remember that that's a major source of nitrogen input into the local waters.
Yeah.
So you would imagine that the closer you are to a wastewater treatment plant, the higher the levels of nitrogen that you might measure, because it hasn't really diluted. It hasn't been diluted yet by mixing with all the other water that's already in the bay.
Right.
So that's one important point. And then why might it vary from one time of the year to another is because remember that that biology is happening, right? If there's nutrients in the water, when things are growing, they're going to be taking those nutrients out of the water and perhaps lowering the concentrations of ammonia and nitrate. Similarly, late in the summer, when all that stuff is dying and decaying, it turns a lot of that organic material back into the basic building blocks of life. It turns it back into the nutrients. And so you might see low nutrients at the peak of growing season because they're all inside the organisms now, and you might see higher nutrients at the end of the growing season when everything's kind of decaying and returning all those basic building blocks of life back into the water, right? And I'm going to show you some data from Jamaica Bay that can help illustrate that, right? So this is a simple little, well, not simple, it's actually quite complicated to put together. But what we did is the New York City Department of Environmental Protection routinely measures water quality all around the city. And we took just the data from Jamaica Bay from 1908 to 2017, and we put it all into a database. And for a 10-year section of that, we created some very basic ways to visualize the data. So it's just ways to sort of explore the data and see kind of what it looks like. And so I'm going to explore the nitrogen data in two parts of Jamaica Bay. The first site I'm going to show you is from this area called Grassy Bay, which is in the deep interior of the bay. There's a major wastewater treatment plant over here by JFK Airport. There's another one over here up in Spring Creek. And they're discharging significant amounts of treated wastewater into Jamaica Bay. So I'm just going to click on this one point, and I get this little data exploration window. And I can do some quick charting. So I'm just going to click on charts, and I can select up to 2 water quality parameters to explore. So because we've been talking about nitrate and nitrite, and we've been talking about ammonia, we'll look at those 2. Okay, and what this is going to do is I'm going to plot a time series. So I'm going to look at those measurements, and I should stop for a second and say the DEP collects these measurements weekly during the summer and monthly during the winter. So let's just see what ammonia and nitrate look like in Grassy Bay. So here we go. So this is are you guys seeing the graph? I just want to make sure you're seeing what I'm seeing. Okay, great. So this is a graph showing the top one is the nitrate and nitrite. Those are usually measured as together, just because of the way that the colorimetrics work. So this is the nitrate and nitrite over a 10-year period on the top, so running from 2001 through 2012. And on the bottom, we have ammonia concentration in milligrams per liter. So the first thing you should always, if I was doing this with my students, I think the first thing I would want them to do is just orient themselves to the axes and the ranges that we're seeing, right? So so if we look at this, what I see right away is that the nitrate values typically range from 0 to 1 milligrams per liter, right? 0 to 1 milligrams per liter. There's a few exceptions. There's this high value right here on December 19th, 2007, we're up to 1.09. But generally speaking, 0 to 1 is the typical range. And Heather, this gets to your question now, right? Because if your students took a measurement and they wanted to know if it was high or low, well, now you have at least one frame of reference, right? Typical values would be between 0 and 1. And so if they got a value of 1.5 or 2 from this location, that would be considered very high, right? And then the ammonium runs from about 0 to 1.5. So actually the concentration of ammonia in these units is typically higher than the nitrate. The second thing that you might notice is that there seems to be a seasonal variation, right? There's almost like a sawtooth pattern. It goes up and down and up and down and up and down. And if I just hover over some of these points, if I hover over the high points for the nitrate, it typically happens in January, right? There's January 30th, 2002. Here's December. So again, it's getting into the winter. This is December. This high point is in December. Actually, so all of these high points seem to be happening in December. It seems to be when you get the highest nitrate readings for nitrate. If I look at ammonia, it's a little more scattered, right? But here the high point is in September. Here the high point is you know, getting into November. This one's September. September. So the peaks, like the highest readings for ammonia, tend to happen in the late summer, early fall. So those are just, you know, basic patterns. And then if I look where the low points are, the low points are happening in the summer, in the peak of the summer, right? Here's July, seems to be when I get the lowest values of nitrate. If I look at the super low values of ammonia, I'm seeing June. I'm seeing, I'm just trying to hover over that one point. I'm seeing June. So they tend to get the low points in the summer. So there's some very distinct patterns that we're observing here, right? So can we explain this? Why would we be getting our lowest readings of nitrate and ammonia in June and July in a place like Jamaica Bay? And I'm going to pause because I would love for it to hear your ideas on that. Like why do you think that our lowest concentrations of nutrients would happen in the middle of summer? Go for it. I don't know who had started to talk, but I'd love to hear your thoughts. You have to think about the biology.
Can we say, like, spring just had happened, there's lots of blooming, lots of organisms are consuming, so now you see a decrease in the amount of nutrients in there.
That's exactly right, Heather. Right, where did all the nutrients go? It's all inside the plant matter and the algae and the organisms that ate that, right?
Yeah.
There's no nutrients in the water because the plants, because everything took them up and grew, right? So this is the time when you would have peak biomasses of things like seaweed out there. And actually, my a former grad student that worked out of my lab had done that. She had gone out and she had measured the biomass of macroalgae out in Jamaica Bay, and she was also taking nutrient measurements. And she could clearly show that as the amount of macroalgae increased, the amount of nutrients decreased, right? Because all the nutrients were going inside the seaweed. So that's exactly right. So then thinking about that, then why would the ammonia go way up again in September? What's happening in September that would make the ammonia, for example, look like it's getting higher?
The algae is dying and the releasing ammonia.
Exactly. That's a great explanation. And typically, when organic matter breaks down, the nitrogen form that's initially released is usually ammonia or ammonia-like compounds. And then eventually, if there's oxygen around, that ammonia will eventually get converted into nitrate. But when you have lots of decaying organic matter and low oxygen conditions like you get in the late summer, you typically find lots of ammonia around. So that's right. And then the nitrate, right? The nitrate doesn't really start to peak until later in the winter, right? When that decaying has sort of slowed down, and a lot of the ammonia is now getting converted directly into nitrate very quickly,
Yeah.
right? And so we start to see peaks in nitrate. If you look at the nutrients a little closer too, you'll see that they actually start to decline again, like really pretty, like usually around March, late February, March, because the early spring blooms of algae really begin, you know, right in February and March. You'll start to see that drawdown already. So that's thinking a little bit about the temporal patterns, right? How things change over the season. And I want to go to another spot in the bay and look and compare. Okay, so we're going to go to another spot in the bay and compare and see and see if things are different. So I would like you to just jot down off to the side that the range of nitrate and nitrite is 0 to 1, and the range of ammonia is 0 to 1.5. And I'm going to close this, and I'm going to close that. And let's go to the far western part of the bay, far away from the wastewater treatment plants of Jamaica Bay. Let's go to this little site right off of Coney Island. It's kind of getting into the open water. And so we would figure that maybe it might be less influenced by the wastewater treatment plants and on all those nitrogen discharges. So let's look at the same thing. Let's look at ammonia and nitrate and nitrite. And let's look at the time series. So we'll submit. And here we go. We have nitrate and nitrite on the top like before, and we have ammonia on the bottom as before. So now I'd like you to just look at those for a second, and you tell me what jumps out at you in comparison to what we saw in the far eastern end of Jamaica Bay.
The ammonia levels are much lower.
Perfect, right? So that's good. The ranges seem to be mostly like 0 to 0.4.
Yeah, yeah.
Perfect. That's one thing. What about the nitrate?
The nitrates are higher.
What was the range that we were seeing at the previous location?
0 to 1?
Yes, that's right.
Yeah.
And now it's 0 to 0.4.
0.2, yeah, 0.4. Yeah.
Well, the range, right? Yeah,
Yeah.
but you're right. It generally speaking, the ranges are now 0 to 0.2 or 0.25 or something. So that's much lower than 0 to 1, right? So we have lower ranges, right? The maximum values are much lower. So that would be consistent, right? That would be consistent with being further away from the major nitrogen source to the bay, which is the wastewater treatment plants, right? So so that's one thing.
Nitrate.
What else do you see? How about the seasonal patterns?
It seems a bit, the nitrates seem a bit more scattered, or the nitrogen levels. It's
Yeah.
not quite as cyclical, or I don't know if that's exactly how you would phrase it, but yeah, that's what it seems. And there's more outlying data as well.
Yeah, yeah. Two great observations, right? Yeah, the pattern isn't quite so pretty. It's still there. If you squinch, you squint your eyes. There's a nice technical term. You know, you can still see that there are some periods, generally speaking, in the summer where the nutrients, right, where the nutrients seem to be the lowest, and they seem to be generally higher in the fall and early winter, but it's not quite as distinct, right? And that could be for a variety of reasons. So first of all, if I showed you some other data, one thing that you would see is that there's not quite as much, there aren't quite as many photosynthesizers in the waters off of Coney Island as there is in the interior of the bay. It's not quite as much seaweed, and there's not quite as much chlorophyll. And so the biology that drives those seasonal changes is a little weaker. It's a weaker signal. So that's one thing. And I think the other thing is that the waters off of Coney Island are, you know, mixing much more with the, you know, it's mixing with waters coming from the bay. It's mixing with waters coming from the harbor. And so the signal is a little more obscured. That's a little more of a, that's a little more complex kind of understanding, I think, of this. But I think that the very basic thing, right, is that we're further from our nitrogen source. So we're seeing lower concentrations. And lower concentrations mean less biology, and therefore we're seeing a weaker seasonal biological signal. But it's still there. It's still there. It's just weaker, right? So, so again, you know, to your question, Heather, earlier, right? You know, when I said part of addressing a student's question of, is this good or bad? Like this, what I'm seeing, the color, is it good or bad? You know, you need to be able to compare it to something, right? And since there's no standards, there's no numerical standards for water quality, really the only thing you can compare it to is like what's typical. What's typical. And I would say that, you know, in Jamaica Bay, those levels, when they get up to 1 milligram per liter, that creates a lot of seaweed. And so we don't really want to see 1 milligram per liter. We like those lower levels, right? But not 0, because then nothing would grow.
I have a question. What is Jamaica Bay brackish water?
Good question. Jamaica Bay is brackish. Just, you know, it's part of the harbor estuary.
Right.
But the greatest source, you know, it's interesting because prior to European settlement, the urbanization of the watershed, there was a lot of, you know, just sort of natural drainage into the bay, which would maintain those brackish conditions. And now those brackish conditions are maintained mostly by the input of wastewater.
I see.
Yeah. So some people refer to Jamaica Bay as the sewage estuary, right? Because it's
Right.
it's diluted
Because-
with wastewater.
Well, the Hudson is brackish. I mean,
Yes.
that's considered brackish water.
Absolutely.
So what is the difference in your readings in nitrogen and nitrate content in brackish versus salt water when you really get out into the open water?
Yeah, so that's a good question. So usually in brackish systems, you know, and again, you know, there's no absolutes in these explanations, right?
Right.
But typically brackish systems are estuaries, right, where the rivers are discharging to the sea,
Right.
and those rivers are typically, you know, collecting nutrients from all across the watershed, you know, farms and forests, et cetera. Yeah. So brackish systems, because they're typically estuaries, are also typically nutrient rich, even in the absence of urbanization and wastewater, right?
Right. Right.
And then as you start to get out into the open ocean, you know, that influence of the land drainage reduces, and nutrient concentrations are much lower out in the open ocean,
Right.
as it gets saltier, right? So that's like the general pattern, right? And then of course, most of our major cities are on estuaries now, and so we have that additional input of wastewater, which just supercharges the nutrient inputs.
Yep.
But actually, I really like that question though, because where you see that gradient of fresher to saltier water, typically you see gradients in the water quality parameters as well. But that's typically because you're mixing like the high nutrient water from the wastewater treatment plants with the saltier water from offshore, right? And as that mixing happens, we're just diluting the impact of the
Right. Right.
yeah. So yeah, that was a good question. Thanks for asking that, and I hope I answered that okay.
Yeah. Thank you.
Yeah. So we're at the end, and I know Cynthia wanted to. I think she has a survey or an evaluation, and I'm sorry I didn't leave much time for that. So I won't. I was going to ask you all to just give some instant feedback on whether any of what we discussed today might be translatable to your classroom, but I don't know if that's part of your evaluation, Cynthia. Maybe we'll just let them answer there.
Yeah, I think that is a better question geared for Galen, but I will answer Amy about the data sets on ArcGIS. So I think if you attended our ArcGIS sessions, Kristen, our curriculum specialist, went over how to get some of those like water quality data sets, but we like BOP definitely, we have our own temperature and salinity and dissolved oxygen data sets, but I can send out an email with some more of those resources as well.
Also, those all those data from the DEP are available through open data, New York City's open data portal.
Thank you. That's what I was wondering. Because I just wanted to, I did attend some of those sessions and I just wanted to incorporate it all. That way I could really, you know, possibly utilize it in the classroom more.
Yeah.
Because I don't want, if I'm introducing them to one system, I just wanted to see if I could get it all in one place.
I'll say this,
Definitely.
Amy. One of the reasons we created the tool that I just shared with you is a couple of reasons. One is we actually did a lot of data cleaning on the DP data set, and we wanted to focus on, we wanted to make it simpler to access at least the data for Jamaica Bay, right? Because that's my particular focus.
Yeah.
And a simple visualization, but you can get all the same data that I just showed is available through Open Data NYC.
Okay, that's great. Thank you. It looks really great. Don't get me wrong.
Oh no, really good. And I wish we had money to keep working on it too, but you know, there's always that problem.
I relate.
Yeah. All right, it's all yours, Cynthia. Do we do?
Yeah, so I'll just drop the link down for our survey. So thank you all for attending this. So we get a lot of our funding from the National Science Foundation. As part of that, any feedback that you can give us regarding that just helps us continue, you know, getting grants and securing funding for the future for, you know, programs like this. Yes, and if you are not able to finish the survey today, I will also follow up with an email, sending out some of the resources that we went through today and also the survey again. Yeah, thank you.
Thank you.
Yeah, thanks all for your attention. I really appreciate it. I hope you got something useful out of this.
Thank you very much. Nitrogen, that's the
Nitrogen.
that's the take-home message.
All right.
So are we
Yeah,
done?
I think we are all good for today, right? Yes, thank you.
Thank
Bye,
you.
everyone. Have a good night.
Bye-bye.
Be well, be safe.